So Dr. Aydogan Ozcan is here getting set up. He received his PhD from Stanford University in electrical engineering and then was a faculty member at the Harvard Medical School. He joined UCLA in 2007, leading the Bio and Nanophotonics Laboratory in the Department of Electrical Engineering and Bioengineering. He holds 21 issued patents, all of which are licensed, and more than 15 pending patent applications. He's authored and co-authored more than 250 research articles, and he's received numerous awards. I'll just list a few that might be knowable by most of us here. The NSF Career Award, the NIH Director's New Innovator Award, National Geographic Emerging Explorer Award, and Gates Foundation Grand Challenge Award, among many others. So with that, Dr. Ozcan.
Thank you. Thanks. It's a great pleasure to be here and share some of our research on the use of cell phones for specifically telemedicine with an emphasis on global health. At the center of our research is actually the cell phone infrastructure that we have, because now cell phones are literally everywhere. We have close to now 6 billion cell phone subscribers worldwide, and more than 70% of these cell phones are actually being used in developing parts of the world. And that's a fantastic infrastructure, because the cell phones themselves are great in terms of their hardware and software capabilities. Now imagine that you can utilize this existing infrastructure for conducting microanalysis, for microscopy, for cytometry, and for conducting diagnosis. That's exactly the theme of my research at UCLA, where we're trying to provide a new platform that is literally tapping to this resource of the cell phone network itself. And on this slide, you are seeing some of these gadgets that we have for literally converting the cell phone itself into a microscope, into a microanalysis tool. So today I'm going to be talking more about this platform, some of our recent results and what it does and how it works, and briefly tell you some of the promising results that we've got and the future aspects of this work. Before I do so, I'd like to emphasize that this is a platform technology, which we engineers call it as Lucas. It has some unique features that are quite different than conventional microscopes, in the sense that this cell phone that actually is installed on the cell phone itself, or a standalone microscope like this, which is almost like a matchbox size, these are actually lens-free microscopes. And that's quite important, because it gives us the compactness, cost effectiveness, and lightweight all at the same time, without sacrificing resolution or some of the unique aspects of conventional microscopy. On the top of this, these microscopes are high throughput. They can look at a large specimen area and volume, which is quite important, especially for looking at rare signatures of disease. And finally, these microscopes that you see at the background here, they're quite tolerant to misalignment. They work in the hands of PhD students, but once they drop the floor, they continue to work, as it should be in field settings. So then the question is this: if you have a lens-free microscope, if you take a lens out from a microscope design, how can you see a specimen like blood cells, which are tiny, which are only a couple of microns in width? And these microscopes are actually computational, in the sense that they work based on shadows of cells. So what is quite different between your own shadow, which is pitch dark, and a cell's shadow is that cells are semi-transparent to light. So they cast a unique shadow, which we take with these cell phone-based microscopes and process them to create images of these cells. As you can see, different particles, different cells, they cast unique textures of shadows, and they contain a lot if you know how to process these images. You're looking on this slide a whole blood sample, which are mostly red blood cells. These are the shadows of red blood cells. Every now and then you can have some different cell, which is in this case a white blood cell. But what's more powerful here is that these cell shadows are the starting point. We can reconstruct them using computer algorithms which run extremely fast, within a millisecond or so, create images that you normally take with conventional microscopes without the use of the lens or the other bulky components. And it gives us, again, as I emphasized, reduction in weight, compactness, at the same time cost effectiveness, because now we have shrinked a bulky microscope into something that you can attach to the back of your cell phone with less than 50 grams of attachment, and very inexpensive components, less than $10, $20 attachment to the cell phone. So with this platform that I've introduced, We've looked at different specimen, of course, blood cells. These are some signatures, shadow signatures of white blood cells, and how you can take something like this, which to untrained eye doesn't look like anything, but then you can reconstruct and look at the subcellular features of, in this case, some granulocytes. And then you can look at the same, with the same microscope, parasites like E. coli and Giardia and Crypto, and then look for the signature of contamination in blood or in milk or other food sources. I'll touch on to this toward the end of my presentation. Because everything is digital, this is a microscope, but it also works like a cytometer, so you can do blood analysis on this for understanding the density of, for instance, red blood cells, white blood cells, or individual cell size. You can do measurements on these field portable microscopes, which are, again, quite compact. One application that we push this to show the promise is actually monitoring of HIV positive patients for CD4 cell counts. At the same time CD8 cell counts. This is an application which conventional cytometers still provide the gold standard. However, as you can see, they are bulky, and per test, they're kind of costly, on the order of $5 to $10. What we've done is we've used spatial, microfluidic, and nanofluidic technologies where you can, from whole blood, capture specifically and sensitively these CD4s and CD8 lymphocytes, and then image them using these field portable microscopes on a chip, so that you're looking at a technology which is field portable and can process whole blood to look at CD4 and CD8 density. This is actually one of these Lucas images. What you see here, in each one of these spots, you have a spatial antibody island which captures CD4s and CD8s. If you zoom into one of these spots, you will see the cell shadows, which are quickly reconstructed and then counted, giving you the local density of CD4 and CD8. This way, we in this study looked at the ratio of CD4 to CD8 in HIV positive patients and looked for essentially a validation of this technology in terms of a conventional microscope to get the CD4 to CD8 ratio for these patients. And these are some of the initial results which show you the premise of a computational microscope that you can attach to your cell phone, either through a USB cable or using a simple attachment, which would give you specificity and sensitivity of a flow cytometer using spatial microfluidic devices, which again have surface chemistry on them. With the same platform, again, as I've touched, you can look at water-borne parasites, from their shadows, reconstruct images, which nicely match it to a conventional microscope, except that these microscopes have larger field of view and larger volume, so that you can look at even more rare events. Some of the recent higher generation, second generation versions of the same technology, as you show here, it's still field portable. Instead of 50 grams, now this is on the order of 100 grams, but still very cost effective. We've now extended it to see malaria parasites. These are some of the Lucas images for malaria-infected blood cells, and these are some conventional microscope images. The infected cells are shown with these arrows to give you the comparison between the Lucas technology and conventional microscopy. One big advantage, as I was emphasizing, is the field of view. And this relates to malaria greatly because the infection rate, parastemia, is quite low for malaria, on the order of 1% or less. That's why you got to look at a very large area. Fortunately, these microscopes that you see here can look at close to 2 orders of magnitude larger areas. Rather than taking a high-power conventional objective lens and scanning this tiny circle here over many field of views, you can screen the entire field of view in a couple of thousand cells instantaneously with these digital technologies. This image that you see here is on the order of a gigapixel. So it has around 1 billion pixels in it. The same platform could be applied to other pathology needs, such as screening of, for instance, pap smear using the same microscopy platform. So what I've shown you so far was a platform which used computation to bring more advanced functions to the cell phone to conduct microscopy. We've recently extended this work to fluorescence. So we've attached to the back of a cell phone another unit which can do now fluorescent microscopy, which is quite important because fluorescent probes are quite specific and sensitive. So through them you can look for extremely rare signatures of, for instance, bacteria or pathogens. And in this case, the attachment to the cell phone is like this shown here. It's a matchbox size attachment on the order of 35 grams or so, which can also work as a flow cytometer. And using this cell phone fluorescent microscope, you're looking at in this image a very large field of view on the order of a centimeter by a centimeter almost. And these are some of the white blood cells imaged in whole blood, and these are Giardia parasites, Giardia pathogens that are imaged using the same technology. Rather than capturing a static image, the same attachment can actually capture a movie and look at now a flowing solution where you're counting cells. And in this case, this is a movie captured by the cell phone where everything that is circled that's flowing is actually a white blood cell, which is a demonstration of flow cytometry on the cell phone using these inexpensive attachments, which can then give you essentially the count of the white blood cell of the patients using the same attachment. And recently, very recently, we've extended the same platform to spot E. coli particles using a very similar attachment to the cell phone. And in terms of sensitivity for instance, skim milk and water, we were down to around 5 to 10 CFUs, colony-forming units per milliliter, which is quite good for a field portable technology like this. And recently, we've extended our work around the cell phone from microscopy, from microanalysis and flow cytometry to literally digital reading of rapid diagnostic tests. And as you know, rapid diagnostic tests come in different flavors. You can literally have hundreds of different types of rapid diagnostic tests for HIV, malaria, TB, or other sexually transmitted diseases, where literally there is a control line and some positive, negative lines that you have to read. What we've created is actually an attachment to the same cell phone, a smartphone in this case, where it's universally going to read different types of rapid diagnostic tests without the human user interfering and trying to read the control line or anything. And we've shown that this is actually more sensitive than the eye because there's a spatial attachment to the cell phone. So let me demonstrate you how this works with my webcam here. I'll just switch to the webcam here, and you can just focus to my hand. Hope you can see. So this is actually, if you focus to my hand, this is actually the conventional Android phone, which has a spatial attachment at the back, and this is where you load your rapid diagnostic tests. What I have in here is a malaria test. It's positive, and we're going to read that automatically and send it to a server for essentially recording that digitally at a Google Maps interface. So you insert it, and there's an application running on the Android phone. So I'll try to show as much as I can. So we inserted the rapid diagnostic test, and this is the interface for the application. Then you press a new test, and you select malaria in this case. And this is the strip that is being imaged in real time. So it's green light because green light is more sensitive, so it's better than your eye, and it's robust in the sense that if your hand shakes or if the sunlight disappears or a secondary light source comes, it doesn't get misaligned or anything. So it's foolproof. All the user needs to do is press on it. Now it's captured this image, processed using the cell phone's processor, and analyzed if it was a valid test, if it was positive or negative. So you can't probably see, or maybe barely see it, but there is a report of the results which show you that it's positive in this case, and it's a valid test. And you can enter some more text about the patient, and then upload this to a central server, which again is like a Google Maps interface, and then you can try this again with different tests. It's not just for malaria. Again, I like to emphasize that it is a universal test, and that's the whole purpose of this, because down the line we will be able to collect information from all these rapid diagnostic tests and display them using the same interface so that you can look at spatiotemporal patterns of disease and how things are spreading over the period of maybe a year or more. And these are some of the screenshots of the same application that I've shown you. You can log in. This is exactly the same test, and this is the report, diagnosis report, and then this is the Google Maps interface, which you can visualize using your Android phone or iPhone or a web interface. And it's shown to be quite sensitive, better than the eye. So All these gadgets around the cell phone are providing us unique opportunities because using these digital technologies, which are now working everywhere in literally billions, we tap into what I call as the micro-internet, in the sense that this transformation of the cell phone itself reminds me of the transformation from PCs to the internet. As we had the PCs, personal computers, cost-effective and compact, we had consumers in millions purchase them and form a network which is quite rich than it was initially designed for. And that's going to be the same thing with these microanalysis tools, with these diagnostic tools running at the back of cell phones or connected to the cell phones. Because of this connectivity and the cost effectiveness, we're going to soon increase the number of personal microscope users, personal flow cytometer users by several orders of magnitude. And this is going to be giving us unique opportunities because the amount of data from the micro and nano scale that we'll be extracting will be enormous. So it's going to give us the big data about the micro scale, about pathogens, about diseases, but then the question is, are we ready for essentially extracting the useful patterns from these devices and the data that they provide? So I'll finish with some final bits of what we've done to tackle this problem, which is the problem of the fact that the devices improve in our abilities to look at these microscale things, whether it's a blood cell or a pap smear. However, technicians or trained personnel who are going to make useful predictions out of these devices are not scaling up with the technology. We can't really have trained personnel in every village in Africa, even though we can have one of these microscopes installed in every village. That's why there's a mismatch between the medical personnel and these gadgets. That's where I believe some smart technologies can come in play, where we can potentially, for instance, create digital superpathologists that never make mistake, even though individually the elements of that network can make mistake. And that's where I think some of our recent work is pointing to. And I'd like you to check this website, which is actually biogames.ee.ucla.edu. If you type biogames, UCLA, you would be seeing it. It's actually a platform which you can crowdsource microscopic images to individuals, whether they are experts or non-experts, in the form of a game, entertaining digital games. And you can collect the responses from these individuals, even though individuals make frequent mistakes, just like poorly trained experts. You can have mathematical algorithms that correct for their errors and make a decision which rarely goes wrong if you have these people working for you. So this platform has been actually out there. You can register for it. These are some of the registered users of this BioGames interface. We have more than 2,000 users playing these games, in this case for diagnosing malaria, and we have so far hit more than 1.5 million cells diagnosed with these gamers. And the bottom line was that they come close to the expert pathologists in their accuracy if you combine the decisions of 10 or 20 of these gamers together, even if they're not trained. So with this, I'd like to conclude and acknowledge all of our funding agencies, and thank you very much.
So we can take one or two questions for a couple minutes, and then we can take more at the end of the session. Anybody has any burning questions right now for Dr. Ozcan? Here comes somebody.
I'm Dr. Vijay Kandula. I'm from the University of Utah. In the last few slides, you talked about a lot of data being gathered and exchanged between different places. I'm sure you would have considered about interoperability and using standards like HL7 and bioinformatics standards. Can you please comment on that?
Are you referring to the security of the data?
No, not security, the interoperability, because data flows from one system to another.
So, I mean, our work is not aiming to create standards that can make different modules go, as side by side to form a bigger system. We're targeting, I think, the initial bits of something that later on would be quite useful. But other researchers are tackling, actually Deborah Esther, and I wish she was here, she's exactly targeting that, which is creating an open source platform where things are shared, standards are shared, so that if I develop this tool and some other tool is necessary for it to really solve something practical, somebody else will know exactly my standards and will be able to write codes for that. So that's, I think, quite important, and I think we have a lot of lessons to learn from Android for the same matter.
Thank you.
Hi, Marion McNabb from Pathfinder International. I'm extremely impressed with this technology. I think it's fantastic. My question is, where has it been deployed so far? And is it to be used for laboratory technicians or for what level of a healthcare worker?
So in terms of tests, we've done tests at UCLA with patients and with anonymous samples. We've done a field visit in South America, and we are going to have some more in Los Angeles for sexually transmitted diseases, and hopefully in South Africa as a follow-up, pending funding. And in terms of the user, it really depends, but most of these things are requiring some sample preparation, and that's where I think they will be used by healthcare professionals who can handle blood or other bodily fluids. So the initial target will be really experts who will be probably handling these cell phones and the attachments with some gloves and with some protocols as before with their other conventional technologies. But there are certain applications of the same platform for the home where you don't require an expert. And one application would be monitoring of quality of water. Even for developed country applications, there is a need for technology to monitor the quality of water, because even in the United States we have 40 million houses with their own wells. And of course in global health this is a much bigger need.
Okay, so we'll take one more question now, and then we can have more at the end.
I'm just a tech guy, but I just had a quick question because we're kind of fascinated with this technology that you just showed us. How much is it going to cost in the future, do you think?
So the cost question is, of course, it depends on the business model of the company aiming to commercialize this. But I can comment on faithfully as the inventor of these technologies on the cost to manufacture or on the cost to put together one of these devices. For instance, the one that I just demoed, this rapid diagnostic test reader, it will probably be less than $20 to create this thing, excluding the cell phone. And the cell phone itself, we value because the second-hand market for the cell phones is going to be enough to get this almost free, the cell phone part.
Oh, all right, thanks.
Can I just squeeze in one quick question? Yeah, so with great technology, with regards to the cell phone itself, are there minimum specifications you need for the cell phone, either in terms of magnification or others? Because I can see there's definitely differences in types of phones.
We work with literally around 10 or more different cell phone types. We're fine. We like Android and the recent ones because their processors are good. But as far as the camera module of the cell phone is concerned, anything above 3 megapixels would be fine for us. So 3 megapixels and higher, they're good technologies, fairly good technologies. And most of our stuff is using actually 5 megapixel, not your iPhone. iPhone has 8 megapixel these days.
Great. Thank you so much for your questions and for the presentation. I'm really excited.